pleasure to be here, and thanks for the invitation. And it's actually a real pleasure to be here to present on behalf of the Alternative Host Research Consortia. Many of our members are here represented. This has been a collective effort to really think about how do we change manufacturing of recombinant proteins in biopharmaceuticals and increasingly food proteins to be affordable, sustainable, and predictable. So how do we go fast in development? How do we break down these barriers? We want to talk a little bit today about the studies that we've done to think about this in the context of monoclonal antibodies as a proxy for other complex proteins. These are heterodimers that have post-translational modifications that require assembly and folding, all the things that Pichia doesn't really like to do, the same way it maybe likes to make antibodies. So this has been a good proxy to think about this question. What I want to do today is take us through some of our learnings and some of the things that we're doing to think about both the host and the molecule to make this class much more accessible. So many of you are probably familiar, maybe you're not if you're deep into fermentation, the current state of the art in manufacturing for CHO-based production of anything with an FC domain. We just heard a really nice talk about this and how it's the current installed infrastructure. Nimble, along with many of the companies that are interested in this space, have worked over the last 10 years to develop a fully continuous drug substance manufacturing process. This process requires the production of material in perfusion. It allows for a continuous feed stream that is free of the cells that then goes through a number of different steps, a capture step that's mediated by protein A. This is actually quite a remarkable element in terms of thinking about how to take that first step in manufacturing post recovery to be able to clear out the materials that are left from the host cells and such. Protein A is a really remarkable material. It also happens to be the most expensive part in manufacturing as a single component by scale. You might have in a manufacturing facility a $15 million column in that packing, so you want to get that right. As we've heard about also, virus inactivation is really critical in mammalian cells, so you need steps for this in continuous processing and polishing, and then some additional reduction of filtration of viruses. So this is what is considered the state of the art today in manufacturing. Volumetric productivity, space-time yield, how we want to think about that, 2 to 4 grams per liter per day of bioreactor fluid of monoclonal antibodies. This is a good number to keep in our mind because once you get past that, chromatography becomes the rate-limiting step in cost reduction. So if we think about food proteins in places where we may need even larger volumetric productivities, we will need simpler downstream processes. So within the consortium, we've been thinking a lot about what does it look like to think differently about this. The consortium was started based on learnings in 2015, 2016 with Biogen and the Gates Foundation, thinking about where are there knobs to change the equation on cost and accessibility and ultimately sustainability. And as we've heard here, also resiliency. Maybe not a surprise to this audience, the secret's in the biology. The choices that you make there really matter, and how you use the biology is going to be dependent on what you have to do downstream to clear things out. And so we and others kind of started to think about this question, and that diagram I showed you before is quite simple, but it can be even simpler when we consider a host like Pichia. If we think about what it takes to stand up, we heard really nicely the time scale for being able to bring a vial into fed batch in CHO. But we know that with the growth rates of Pichia is much faster, and so we can get away with a little bit shorter seed trains. There's examples in regulatory filings now of allowances without having to have viral inactivation. These turn out to be pretty expensive steps in kind of two ways. One, how many people like ceviche? I think we're having tacos or something for lunch. Ceviche is made by adding acid to your protein for a prolonged time to kill off whatever's in there. That's also what they do to inactivate viruses in the recovery steps in this. So you have to get the timing of that just right in order to not turn your monoclonal antibody into aggregated material. And then I think the real question is, could we get rid of protein A? And several things that we've done over the last several years, and some work that was done by one of our founding members, Amgen, to look at these questions, and recognize that based on some of the literature and some of the work that was done previously in the lab, thinking about small footprint manufacturing, we showed that actually you could get rid of protein A and start to platform the downstream processes, not just for monoclonal antibodies, but for a wide range of different proteins. And so we kind of thought about, okay, if you start to get rid of process conditions This is going to save on costs, but then the other thing that's really important here is it also saves in sustainability. So the water reduction and fewer buffers, the HVAC for your facility size, that's the largest energy cost in building a plant today is the HVAC that goes around it. And minimizing waste streams, right? If we switch to single-use systems like the ones we heard about yesterday from Sunflower, these are ways in which we can think about reducing the overall footprint and carbon footprint of these facilities. And so one of our members, actually Sartorius, who's here represented, she was here in the audience somewhere, has done some extensive study, and this is work from Benham Partopu, who, his email's there if you want to reach out to him, has done a really excellent cost analysis comparing that CHO process that I showed you, using standard single-use platforms, a Pichia-based process, and a C1, this is the dyadic fungal host, that's made quite a bit of progress over the last couple of years in its expression of monoclonals, and recent paper out with that host. And what they showed is that going through a kind of full facility build out and the estimations on what you could get to in terms of cost of goods per gram for a monoclonal antibody. I recognize for foods we need like 3 more orders of magnitude here, but let's talk about biopharmaceuticals. That actually for both Pichia and fungal hosts, you can get into the kind of $10 to $30, $40 a gram range, kind of interchangeably between those two, by taking out a couple of these steps and moving to fully continuous downstream operations. But also, wait, there's more. You get to reduce down your plastic work stream. So from a sustainability standpoint, all the benefits that have already been modeled out for CHO get even better because we have fewer total operations. So this looks pretty compelling as a way to think about what future manufacturing states could be using this. And they took this one step further and just kind of showed overall as you kind of look at intensification that you start to intensify your process and making more and more doses with Pichia and a continuous process that actually can be more efficient in your facility utilization than CHO, and that the equivalent operating output of CO2 is less comparatively to a CHO-based process. So the question is how do we enable this as a future state in biomanufacturing? We have to build out some of the process equipment, we have to understand how to intensify these things, we have to build better strains, we have to learn how to produce the proteins appropriately. And this is really where the Alternative Host Consortium comes in. It was founded in 2018 as a way to think about how do we systematically establish the ground rules for engineering proteins and hosts such that there are parts and tools and kits and protocols so that everyone can go out and do the things that they want to do to make the proteins that will save the world. And so in doing so, we've been thinking a lot about all the critical parts, many of which we've touched on in this meeting already here today. What are the base chassis? What are the vectors? What are the promoter sequences? How do we think about the molecule itself? What about all that pesky post-translational modification stuff? We have to address all of those different elements. And so our consortium has been growing over time as a way to try to address this question. It operates in an open source, pre-competitive way. There's some side benefits of being hosted at MIT with respect to things like gene editing tools I can tell you offline. And what we're trying to achieve is a set of places for everyone to start to do the work that they need to be able to do. And so what I want to share with you today is just some of the things that we've been doing. Many of you have probably seen some of these papers. I bring it up here just simply to say that one of the first things you have to do is you have to be able to read and write the genome. We did this very intentionally. We went out and sequenced a number of different banked strains, reported those differences that were out there, and then used that to kind of think about what are kind of critical changes that might be necessary. And then excellent work in the kind of CRISPR-Cas9 space across the community, but one of the things that we recognized was that a lot of the efforts that have been out there used a different cut site and processing of the tRNAs. So we went and did a bunch of sequencing to see how Pichia natively slices up its mRNAs and found some interesting Pol3-based promoter strengths that allow us to create multi-genic knockouts or cut sites in the same host. So kind of published on that, and so now we have a standard tool, what we call the ALTUS, and I've noted here some of the base strains and things that might be of general interest here. The R vector here is our tool for using for that purpose. All right, so let's talk about monoclonal antibodies. If I want to make it for biopharmaceutical use, I have to think about the critical quality attributes. Some of these are known, and some of those, as we just heard, some of them need challenged as we think about this from a clinical experience. But things that are going to kind of be on the minds of folks as they think about this is, what are the variations in the sequence? Do we have inter-C-terminal variations? Glycosylation, that's a big one in this community. Charge variance, what do we do about full length versus partial products on the process side? And ultimately, what is the overall yield, right? And space-time yields matter, cost matters. No one's gonna replace CHO unless you can do much better. Interestingly though, Amgen showed that 50% titers out of a Pichia process are cost equivalent to a CHO process. So we don't have to be that much better than CHO, we just have to be in the ballpark. All right, so to do this, we started at the beginning. We have a common tool we use in the consortium called the L-Vector. It's a Gibson-based assembly structure that allows you to mix and match parts, a number of different antibiotic markers on it, or selection markers for the yeast strains. We created a dual expression system for producing trastuzumab and a bunch of different other antibodies. We liked this as a concept because we know that things like copy number and the ratio of heavy and light chain is going to matter. This is known in CHO and some other hosts. So we wanted to be able to play around with that, and kind of generally we've kind of worked towards optimizing that ratio and such. We have an additional vector now that's called the A vector. I don't know where they name all these from, but the A vector has both of them on a bisostronic construct. And so the other data I will show with you is the optimized version of that. One thing we like to use is we use the DOS2 promoter quite a bit. That turns out to be a pretty nice one. We also like that as an integration site for single copy integrations, or for single site integrations. All right, so if you have to know your enemy. What are you up against? We're up against Pichia trying to do bad things to monoclonal antibodies. We wanted to characterize what does it do natively. So we made trastuzumab, and we've done a whole bunch of different analytics. I'm kind of summarizing a few of them here. These are, you can see the gel on the one side, kind of different elements that are labeled there based on intact mass spec data, and a little bit of peptide mapping we've done as well. A couple of things you kind of appreciate. We're using the alpha mated factor with pre-OST pro alpha construct. We've tested some other ones. They all kind of work kind of similarly in our hands for IgG. You see a little bit of poor cleavage of that N-terminus, especially on the light chain, but a little bit of evidence of the heavy chain. N-link glycosylation variation, yeah, that's a known issue. A little bit of O-link monosylation. And then an FC cleaved product. Again, all things that are kind of known generally, but as a starting point, these are the things that we have to pay attention to. So what we did was we took all those data and we went back and looked at the sequence itself, and then we started to ask, like, what are the most conservative changes we could make that would be within the realm of clinical experience and human experience that would actually make these antibodies by sequence more productive in Pichia. And so there's a couple of things you can start to highlight. You see our favorite KEX2 sites, the glycosylation site. There's this FC cleavage site that's in the middle. Actually, the FD that was in the last talk at Tenzimab, not surprisingly, there's some Pichia experts in the room that were involved in this, got rid of that particular site. They put in this AR dibasic motif. So again, clinically validated motif. There's actually a number of clinical antibodies from a couple of companies in Japan that also use it. They make it in CHO, but nice example. And then this cleavage problem on the N-terminus, one thing that bacteria do when they translate everything is they put a methionine on the front. So there's already experience of single amino acid variation at that first terminal spot. So we said, well, what's the shortest we could cut down the EA repeat structure that's in the canonical alpha mating factor between the KEX2 and the C13 site? We've previously shown that little spinach is hard to get to sometimes, depending on how accessible your N-terminus is in the fold. And so a single glutamic acid nicely cleans up all of that at that one particular site. And then you can modify the N-link site to make it a glycosylate, as we heard in the prior talk. Interestingly, a lot of pipelines now, because people don't want the extra function that comes from that FC domain or the variability that comes in CHO-based manufacturing of the glycans, folks are starting to engineer those out in a variety of different ways in pipelines. So does it help? Yes. This is a whole bunch of different commercial antibodies that we've now made and expressed, and you can see, in many cases moving towards 99.9% of pure heavy chain being produced with these two modifications. The Genentech antibodies and Eptinzumab are actually the best, so there's some fine nuances there still to try to understand, but something about those structures. We try to make conservative changes, so we're picking things that are apparent in the human genome and other banked sequences. The good news is there's now lots of databases of banked antibodies from humans that give us ways to think about making those changes. We had a question, some of our members at Biogen said, well, this is cool, could we make it in CHO? Like, could we think about interchangeability? Like, let's go fast to the clinic and test the Pichia antibody, but then it's a well-defined molecule. Could we just make it in CHO later with our other facility? So we said, okay, great, so let's take, in this case we did NISTMAB, which is a standard that Nimble has put forward. It's an AZ molecule. It's not a very great one. It binds RHB. It's not the best expressor. It's not as good as Herceptin or some of the other ones that are out there, but it expresses okay. We made the same changes in both the yeast and the CHO, and you can see here in the gel, the two ones with this particular change, very comparable heavy and light chain. We did mass spec. Again, you can see a little bit of the mannose ringing in there, if you will, in the mass spec. These are like 1, 2, 3, 4, 5 mannoses that are extended out. So we heard there's already kind of clinical experience with these. We find that varies with antibody. So I think this is more a mark of poor folding or slower folding translation in the cells. Interestingly, we don't see the C-terminal glycine clipping, which is a really common variant in CHO. So that's kind of an interesting fact. Anecdotally, this variant also expressed better in CHO, in fed batch cultivations at small scale. I can't say much about that, but that was kind of cool, side note. And then circular dichroism, you can see that both the yeast and the CHO-based material are nearly identical in their secondary structures. So with this, we felt really good. Okay, maybe we're starting to learn some rules about how do we actually adapt the molecule to be made effectively in Pichia with things that are effective. And the question now becomes okay, can we make more of it? And so, we've been thinking a lot about this question for a while now, is how do we supercharge K-FAFI? And we've heard a lot of really great examples. We want to do this systematically, obliterate it, find all the ways, and just put it out there that we can figure out how to do this efficiently. Because there's a lot of things that need to get done, and we need some different ideas than the things that have been out there. So we've been interested in engineering the secretory pathway and the metabolic pathway, both. I thought I'd give you just a couple of highlights of things we're doing there. Some really excellent models for GEMs, genetic genome scale models that you guys have built. We've been playing with the most recent V3 version. One of the things that version did that was nice was it added in all the metabolism around methanol and some of the other nuances of amino acids. So we've been building on top of that, adding in pathways and reactions for translation and secretion. And here we've used trastuzumab as a model. And then doing some analyses to look at changing the optimization function from growth to production. We've been able to nominate a whole set of genes that we want to add in an extra copy of based on these predictions to see if it can help improve and reduce metabolic burden. We've been testing those a couple of different scales. I think they're on tap to go into Ambers very shortly. Just some of the data, here's a whole bunch of genes that were predicted based on the model. I mean, I think the models aren't perfect yet, and we're still trying to figure out how to improve our prediction capabilities given the molecule of interest. But in small scale, these are 3 mil cultures, several hits, quote unquote. We don't really believe hits until we get them into flasks or reactors. But a couple of interesting things just based on the talks the last couple of days. More carrier proteins for ADP, ATP, more NADH dehydrogenases, right? These all sound good. Needs more energy, and in fact, these work nicely at 50 mil scales, so we're now seeing titers over 50 mil shake flasks in kind of a fed batch model, about 100 mg per liter, almost double to triple what we're seeing in the, so this is one gene copy, one site in the genome added, so very, very minimal changes. The answer's not the same for everything. I throw this in here just as a cautionary tale. Don't take those and go run out with them. This is HSA. You can see some of the same predictions that are in there, but actually some of the other genes are more important. So I think your mileage may vary a little bit depending on what the protein is that you're interested in. But we think this is going to be a generally useful way to ask this question. The other thing we've been doing is, CRISPR is a great tool for learning about how the organism responds to perturbations, and you can do this at a genome-wide scale. So we did a full essentiality screen across the organism. The way this was done was we had strains. We built a library of guides targeting every gene in the genome at 5 different sites. So that was a total library. Every gene, 5 different guides, because they don't all work. And then we plated them, or we transformed them into strains that either constitutively expressed Cas9 natively or did not. And then based on that, we sequenced both pools. We scraped the plates and sequenced everything off next generation sequencing, counted all of those, and then we developed a scoring algorithm for essentiality based on the presence or absence of scores in each of those. And so we now have a full catalog of all what we would call essential genes in there. You can decide what you want to call that cutoff. We have an internal mark of about 0.3 on this score line that we say below that is non-essential. So if you look at a family like cell wall genes, you can target knockouts that are in that. These should be relatively safe under these kind of slow growth conditions that we've done the selection and screening at. So we can knock them out, test them, see if it helps. This is just the total set by families. A couple of interesting things, about half the genome is within range of targeting, so that's kind of nice. That gives us a lot of options. These are single knockouts, so we don't know exactly the extent to which you can put all of these together. But in work that we've done in kind of looking at targeted knockouts in the secretome based on essentiality, it took us up to 11 hits before we started to see real reductions in the overall stability and growth of those particular strains. So rather than just kind of randomly picking things or picking things that we thought were good based on pathways where we had a hard time knocking them out, this sort of guided approach using essentiality was important. A couple of things, translation, maybe not surprisingly, has the lowest percentage of what we would say is non-essential. But everything else is kind of in that same range. Stress responses, I guess, depending on what kind of stress you're talking about. Remember, we've screened these under a plate condition, so some of those may turn out to be more important in a reactor. But we've been systematically trying to go through this to validate that this idea will work. We're working on ways to scale this up to the whole genome and by many pathways now, but looking at this, we picked two families, cell wall and vacuolar. They're both involved in transport. And we said, look, there's some targets in here. And you start to look in these families, and there's some things that are reported in the literature as prior precedents as being important. So we went and knocked those guys out. This is now producing IgG1. And you see kind of a nice, at small scale, 1.5 to 2x, and maybe even up to high levels of titers against our base strains. These are all done at 3 mil scales, so we want to be able to do it at a larger scale. The other thing to note here is that colonies here, they do have a slightly different morphological structure, but all of the strains that I will show you here have been in the reactors now. So this is probably our best single hit on the strains for mAbs. I mean, that's 2.5-fold there for this JSN1. It's worked really nicely in reactors. The XG1 also tends to be pretty consistent. CD8-2, interestingly, is a hit that works really nicely for HSA, but not for IgG1. So again, I think there's going to be some learnings across classes and chassis of what we have to build out. In the vacuolar transport space, a couple of them, some of our favorites. I think this is our current favorite one of that set. We're starting to do things to look at these in pairwise and other interactions, and looking to scale this at the genome level to be able to do it across many different pathways. And then I thought I would just end with, we've been talking a lot about machine learning, just one more thing we've been thinking about on this front and building tools that are helpful for members is really thinking about how do we improve codon optimization. I think everybody's had the experience of codon optimizing things and it's not really working better, and you put the native gene in and it works really nicely, what, right? Why do these tools not work better? Expert in the lab, Narinian, who's a postdoc, is a deep learning expert and has been thinking about this question, and so she's been developing natural language models to take proteins and convert them on a species-specific syntax into coding information. And then you can mix and match these based on the direction you want to take. If you have a human, you can translate it back into Pichia or Chode, you can go whichever direction you want. This is still kind of early data, but what we're finding is that these are much more consistent in the overall improvements or the specific productivities we get out versus some of the other commercially useful tools that are out there. Again, it's not a panacea, it's not going to fix everything. We have to address many of these different elements to get the best performing strains, but what we want is consistency. We want to be able to engineer these things purposefully. So we think these kinds of tools will be helpful in that regard too. So I'll just finish here. I know it's lunchtime, people are probably hungry. I've talked about 3 things, and I think actually all 3 are critical to solving this problem. We have to think about the molecules that we're making. How do we make conservative sequence changes that are going to be acceptable and we can get clinical proof around to show that these are actually tolerable both as medicines and as food proteins, but that can be made easily in yeast organisms. So we need to not be afraid of that particular part of the question. On the strain engineering side, I showed you kind of two approaches that we're thinking about this. I think there's other ways to think about gain of function as well, but it's a little bit more tricky. We think that actually taking things away opens up more space generally metabolically, so we're kind of favoring that as an approach right now. And then on the process engineering side, I didn't have time to talk to you today about some interesting work we're doing in active learning strategies to improve media formulations to cut designer experiments down by 3 to 5-fold. So you can go from like 1,000 experiments you have to do to like 50. It's pretty cool, works really nicely. And I think it's gonna help us figure out how to address some of the deficiencies in the media formulations that are necessary to sustain super high viable cell culture densities. We saw yesterday in the talk with the sunflower system, the wet cell weights of like 700. That's a really high wet cell weight. And to be able to maintain that productivity and to push them to be able to continue to produce, we're gonna need different media formulations. That's a learning that's come from the CHO community. And I'll just stop there, and I'll be happy to answer a couple of questions. These are our current members. If you'd like to know more about the Altos Consortia, please feel free to reach out as well. Thank you.
Thank you very much for the nice presentation. I had a question to the strain engineering part, specifically the metabolic engineering. Which promoter were you using for the different proteins? Did you look at different promoters? And specifically, you're looking at the secreted titers, and with some of the metabolic engineering strategies that you looked at, would they have also affected the promoter strength, the expression level of the transcript itself? And yes.
Yeah, so we've just gotten back some of the transcript data on those. Those are relatively fresh studies. The transgenes still express at a similar level. In terms of promoters, we tend to use DOS2 and AX1 just as an initial screening tool. We find that they're just consistent, and we want to be able to look things across to be able to make fair comparisons across everything. So we don't. Our experience actually has been that we find that promoters are important, but not that important, right? They're actually getting it in the right place in the genome, finding the right way to integrate the right copy number. That can be more important than promoters. But it is a bit anecdotal still, and we're still kind of learning as we go here.
I believe there's a guy, Anton. Yeah, thank you. A fascinating talk, thank you. Fascinating speed and broad tools. One question, in the first part you mentioned polymerase III promoters for the CRISPR for multiplexing. So did you ever sequence the strains which you obtain? Because when we sequence strains of multiplexing, the whole genome is messed up because due to Pichia non-homologous recombination, we get all different chimeric forms of chromosomes, but never the mutations we want to have.
Yeah, so we tend not to use the multi-gene copy when we're making chassis. When we're building chassis, we do them individually and sequentially, but when we're doing screening, we often do kind of multi-genic screenings.